Good day, my name is Stoychev. I'm a senior researcher at the Molecular Diagnostics and Omics Group at the CSIR, where for the past 12 years we've been developing workflows for biological mass spectrometry. I'm also currently the head of Proteomics at ReSyn Biosciences, and our focus is on implementing sample preparation workflow again for biological mass spectrometry using our patented microparticle technology. Today I want to give you a brief overview of how Proteomics is entering the clinical research space and how we are starting to apply it within a South African setting. When it comes to health, one of the global challenges for the 21st century is to be able to more precisely diagnose disease as well as predict its course, and further to be able to ensure more effective treatments. However, the current status quo is rather different. For every patient that is administered with a certain drug, there are many others that have no response or have a negative outcome of that treatment. In South Africa, it is estimated that close to 12 billion rand is spent yearly on treating the side effects of various drug treatments. In addition, we are not very good at picking up diseases early on, and this a good example is the various cancers, where often we only detect these when late symptoms occur, and by this time, the chances of patient survival are quite low. So rather than applying a blanket approach, we require a healthcare system that is tailored to the individual or stratified portion of the population. And this is where precision medicine comes in. It integrates different layers of information about the patient's genes, proteins, clinical data, environment, and so on in order to be able to diagnose and treat disease more accurately. So in this particular presentation, I will focus more on the novel omics-based technologies such as proteomics and how we plan to apply this within the clinical space. Now, before going into and describing proteomics and how it's applied within clinical research, I would like to highlight some of the developments within the field of genomics and genome sequencing, since there are a lot of parallels with what is currently happening with proteomics. So back in 2000, President Clinton announced the completion of the first survey of the entire human genome. And as we all know, our genome is essentially the instruction manual and is present in almost every living cell. Now, understanding and being able to read this sequence allows clinicians to better predict and understand the predisposition of patients towards diseases. Now, the completion of the Human Genome Project took approximately 13 years and at a cost of about $3 billion. Currently, in 2012, we can sequence a human genome at an approximate cost of $1,000 to $5,000 in about 2 days. Now, I would like to highlight some of the major drivers that have been responsible for reducing the cost and the time. Because you'll see in subsequent slides, there's a lot of parallels with what's currently happening in proteomics. So standardized workflows, high-throughput sample preparation, as well as the evolution of sequencing technology has been critical in reducing time and cost. In addition, developments within computational assembly and reference sequences or databases has also been important in reducing time and cost. So essentially, these drivers and these developments are now allowing for genome sequencing to start to be integrated within the healthcare systems across the world. A good example is the 100,000 Genome Project, which was conceptualized based in 2012, and essentially integrates genome sequencing with medical records of patients within the UK National Health Care System, and allows clinicians to be able to better understand the predisposition of patients toward certain rare diseases and cancers. So the way genome sequencing helps us to predict disease better is be able to read out the type of genes and their variants or mutations within a patient sequence that either have unfavorable or favorable outcomes when it comes to different types of diseases. However, genomics on its own will not provide us with all the answers. It is well understood that it will not tell us when the disease will occur, how it will progress, and what is the effect of the treatment in real time. And this is where proteomics can add real value. So the proteome is defined as a full collection of proteins characterized in a holistic manner for a specific system at a given time. And as we all know, proteins are the products of DNA, and what's important to understand is that their expression or production is a dynamic process. Their interaction within pathways then drive cellular processes, and in be able to monitor the changes in the cellular processes as well as changes in protein expression allows us to then detect when the disease is occurring. It allows us to understand how it's progressing or its severity, as well as allows us to understand what are the effects of the specific treatment. What's important to also note is that besides the effect of disease on the proteome, it can also change due to non-specific factors such as our age, race, diet, as well as other environmental factors. So it's important to be able to differentiate between these non-specific factors of noise and versus the changes due to disease. And in order to be able to do that, we need to be able to screen large enough numbers of the population, establishing a baseline for each patient, and therefore be able to differentiate between disease changes and non-specific changes. But how difficult is it to measure the proteome? Let's first look at the complexity. It's estimated that we have 20 to 25,000 human genes, but due to alternative splicing, we can end up with almost 100,000 different RNA transcript variants. And when proteins are produced, they can be decorated on the surface using different post-translational modification, leading to up to a possible 1 million different protein isoforms. In addition, proteins are not always, proteins are not present at the same abundances or concentrations. So in some instances, such as plasma, you can have up to 10 orders of magnitude difference between the highest abundant protein and the lowest abundant protein. In addition, as I mentioned, the proteome is dynamic. And so if you want to measure changes due to disease, we need to be able to do that at the correct time and actually longitudinally over time. It's also important that we collect the samples correctly, because if this is not done properly, we would lead to artificial changes that are not associated with the disease. So how do we measure these incredibly complex and dynamic systems? There's numerous ways to do that, but by far the most popular currently is using mass spectrometry. At the very basic level, mass spectrometer can be described as a finely tuned weighing balance. So by measuring the masses of proteins, we can identify their composition. And we can do this very sensitively, down to the attomole levels. We can do it in a wide dynamic range, in up to 5 or 6 orders of magnitude now. And we can measure tens of thousands of proteins within a single sample. So we can do this in parallel without need for serial position. So if you measure a healthy versus a proteome of a healthy person versus one with disease, or if you measure the proteome of a person over time, we can start picking up the minute changes that are associated with disease or disease states. But what are some of the barriers that have prevented proteomics from entering the clinical research space? And if you look at these, you can see a lot of parallels with what was happening within genomics. So our sample prep has been low throughput due to the complexity of the proteome and due to the lack of availability of standardized workflows. Data acquisition has been quite slow, taking up to several hours for single samples. And we struggle to process data efficiently in a timeliness manner. Similarly to genomics, the field of proteomics has been rapidly evolving. And now we can more efficiently profile a proteome than what we can do 5 or even 2 years ago. A lot of the major developments have been upfront in the sample preparation, so getting the sample from the patient to the mass spectrometer. We've been involved quite intimately in this as part of ReSyn Biosciences, where ourselves and others in the fields have found ways to more efficiently integrate different steps and be able to integrate this within high-throughput automated systems in order to improve the robustness and throughput of the sample preparation steps, as well as standardize these steps. So we now have the standardized, rapid, robust proteome profiling pipelines, such as the one shown here, where upfront we've gone from days to hours in terms of sample processing. Further, the mass spec technologies have evolved. They're more sensitive, but they're more importantly also more robust. And we've decreased the analysis per sample significantly from hours to minutes. So this effectively now allows us to process thousands of patients' samples within a timeliness manner. And essentially what we can generate is a digital fingerprint of each patient that will allow us over time to predict their well-being. These developments are fundamentally changing our approaches when it comes to the application of proteomics within the clinical research space. So up to date, we would start off with very few patient numbers during the discovery stage, and we would try to profile as many proteins as possible. We will then increase the number of patients during verification and validation, and typically the validation will be done with a non-spectrometry-based assay as well, which can lead to issues. The major issue, though, with this approach is that we are heavily underpowered during the discovery stage. As I mentioned, the proteome can be affected by many external factors that lead to noise or variability that's not associated with disease. And this can result in many false positives and ultimately to biomarkers that are non-specific or non-selective. And this has little value when it comes to the clinical diagnosis. So due to the improvements in robustness and throughput, we can now start with much higher patient numbers during the discovery and match this during verification and validation. The major advantage of this is then we have more statistical power. In addition, instead of looking at single biomarkers, we're looking at biomarker combinations or protein combinations that leads to protein signatures. These signatures have much higher selectivity and specificity. When we start combining these protein signatures with genome sequencing data, metabolome data, as well as clinical information, we can have much better prediction models that can be applied within the clinic. In order to illustrate how we are applying these developments within the clinical research space, I would like to highlight some of the data we are currently generating for Tenofovir-induced acute kidney injury in HIV patients. As we all know, there's a high burden of HIV infections within South Africa, with close to 8 million people living with the virus. Approximately half of these are on some form of antiretroviral treatment. Tenofovir is part of the first-line treatment. It's a reverse transcriptase inhibitor, and typically is rapidly converted into active metabolite, whose unbound form would be excreted by the kidneys. However, in 8 to 10% of the patients, acute kidney injury is developed. The mechanism is thought to be due to retention in the proximal tubule cells, inhibition of DNA replication, leading to induced cell death and ultimately kidney failure. So at this point, the patients will have to be taken off the treatment, put on dialysis, and if the damage is quite extensive, they could lose the kidneys, and ultimately this can also lead to death. The current tests are rather delayed, and clinical tests are rather delayed and unreliable. They're influenced by a number of non-renal factors such as muscle mass, fluid intake, and diet. So we had two main objectives for this project. The first one is to try and identify predisposition factors that can guide the clinicians to which patients will develop the disease and therefore can be put on different treatment. The other aim of this project was to identify protein signatures that can detect the disease earlier as well as be more precise than in the current clinical tests. The way the study was designed was that patient information as well as samples were collected to clinical sites. Part of the cohort was then screened using quantitative PCR. I'm not going to go into details in the results of this because I want to focus on the proteomics workflow, but essentially from the several hundred variants associated with tenofovir metabolism, a panel of 9 polymorphisms was selected, and this was able to separate the cohort into AKI and non-AKI patients with high confidence. So next, this panel needs to be validated in order to understand if it can be used as a predisposition tool for tenofovir-induced AKI. On the proteomics side, we would go with the rectangular approach as I just described. So we would screen 2 independent cohorts using mass spectrometry, and then we would extract the differentially expressed proteins and see which are overlapping across the 2 cohorts. These will essentially be our candidates that can be inputted into different combinations to form our signature panels. These panels will be further validated using orthogonal assays such as the Illumina amino affinity workflows. Ultimately, we would integrate the omics with clinical data in order to form prediction models that can assist or be integrated with current clinical tests. So let's have a look at how we go about extracting these proteomic signatures from our data. As I mentioned, we would profile the 2 cohorts independently using mass spectrometry. From the first, we managed to quantify just over 1,000 proteins with high confidence across all the patients. Next, we extracted the differentially expressed proteins that were statistically significant across the AKI and non-AKI patients. And this was done at different folds, fold change, 1.5 to 4-fold. Next, we repeated the same process on the second cohort. We would then look at which are common across the two data sets. And what we observe is that even at the most stringent cutoff of 4-fold, there were 13 proteins that were overlapping across the two cohorts. And a more lenient cutoff, but still significant of 2-fold, we have 77 proteins that are common across the two data sets. So this is essentially our verified list of candidates we can then use in different combinations for our protein signatures. So in order to prove the concept, we took only 4 proteins as a panel, as a protein signature panel, and we did a retrospective discriminant analysis. And we can see that there's a very clear separation between the non-AKI and the AKI patients. When we compare this to the current clinical test, we can see there's a lot of overlap between the patients and those that have no AKI. So it's very clear to see that even a simple 4-protein signature can have better discriminative power than the current clinical tests. I would also like to highlight that we have other layers of information available from our data. As I mentioned, proteins interact with each other and drive different cellular pathways. So by functionally annotating differentially expressed proteins within the data sets, we can confirm the mechanism of action behind tenofovir-induced AKI. And this information is really important when it comes to rational drug design studies in the future. We next verified the performance of our 4-protein signature using the orthogonal Luminex immunoaffinity platform. We achieved 97% specificity and 90% selectivity. The follow-on question was, could we improve the performance of this 4-protein signature if we include the clinical variable? And this was very much the case. It must be noted, of course, that we only did this on a relatively modest cohort of 78 patients. And as we do the validation on larger patient numbers, several thousand, the performance will surely drop. However, because we plan to do the validation using unbiased mass spectrometry, we can improve the performance of the model by adding additional verified candidates into our protein signature. This will be done in an iterative manner that allows us to fine-tune the specificity and selectivity of the predictive model in order to achieve the maximum performance. This study is very much ongoing, and as the next steps, we plan to perform the validation study on a much larger cohort of at least 1,000 patients, and this we would like to do across different biofluids such as urine and plasma, in order to be able to expand the number of candidates that can go in our protein signatures. We would also like to perform the validation longitudinally, including patients that are very early on in the treatment, in order to understand if our panels can provide earlier detection than the current clinical tests. The analysis will also be performed on patients that are on other treatments outside of tenofovir in order to be able to understand the broad applicability of the signature panels. Lastly, we would also at some point like to validate our genetic panel and understand if this is good for filtering patients upfront in terms of the type of treatment they need to receive. So before I end, I would like to highlight some of the challenges we've encountered when embarking on some of these clinical proteomic studies, and hopefully initiate a conversation about how to extract the most value and impact out of these cutting-edge omics technologies. Probably the first thing we need to do from our side is prove the value of these technologies a little bit better to the wider community. And in order to do so, it's critical that we're able to complete this type of clinical studies that I've just described and then package the information in such a way that it can be presented in a meaningful way to the various stakeholders, being clinicians, policymakers, as well as the wider non-academic community. The point being is that they should be able to see the value and the impact it can have in the clinical space. A major challenge that we encounter is that of sample quality and availability. Most of the clinical proteomics projects that we've embarked on, we had to start sample collection from zero, and this has obvious implications in terms of the timelines. The issues have been twofold. First of all, samples are not readily available. And when they are available, they are often not suitable for proteome profiling. So we need centralized biobanking facilities that can be accessed and collected the samples in such a way that they can be profiled using the latest omics technologies. Another aspect that needs to be addressed is that of infrastructure and equipment. With proteomics being rapidly developing, we need access to the latest cutting-edge technologies, and we need sufficient capacity of this instrumentation in order to be able to tackle the large-scale population-wide profiling studies that have the most impact. The issue, though, is probably not to the level of infrastructure investment within South Africa, but how and where this infrastructure is being put down on the ground. In reality, what we are seeing is that a lot of the instrumentation is not being utilized fully or not being utilized at all. And probably the major reason for this is lack of knowledge and expertise in order to extract the most value out of this cutting-edge equipment. So the first thing we need to do is increase our skills and expertise within this field. And we also need to relook at our funding models and see if they need some readjustments. Perhaps we need to look at centralized centers of excellence, such as the ones in North America and Europe, where the expertise are concentrated and then the wider community access these. We also need to coordinate our activities a little bit better. So initiatives such as Diplomics and ACGT are very important, since these are focused primarily on being able to coordinate the activities of different omics labs across South Africa and extract the most value out of their offerings. The data that we generate from these omics technologies probably is the most valuable resource that we have. But in order to extract the most value out of it, we need to be able to mine it efficiently. So we need to integrate bioinformatics, statistics, and machine learning skills so that we can then integrate different levels of omics as well as clinical information into our models. We need to be able to address different storage issues and security and access barriers. If this is all done properly, this incredibly information-rich data can be used as the knowledge of our, as the basis of our knowledge-based economy, as well as to grow our biotechnology sector. Because the data contains so much valuable information, novel information, it will generate new ideas, and these will drive novel startups that will expand the biotechnology sector. We also need to understand the level of investment that is required to bring these technologies or the impact of these technologies to the market. There's not a lot that we can gauge in the South African context, specifically in the phase of proteomics. But if you look what's happening overseas, we see significant investment into companies that are focusing on omics and specifically proteomics for clinical applications. So these are just some of the examples, but we can see the level of investment is being made into them. And what we can gauge from these numbers is probably two major things. Firstly, that the technologies are mature enough to justify this level of investment. And secondly, that there's a lot of value in the application of omics and specifically proteomics for clinical applications. What is also clear is that we're going to struggle to raise this kind of funding in the South African context. So we need to be innovative in the way we think about how to take our products and technologies to the market. Lastly, we need to look at policies. Generating such big data sets has its own challenges in terms of ethics, privacy, and confidentiality. We also need to understand how to access data from a commercial perspective. So this conversation needs to happen now so that in several years' time, we can be able to have a clear path from the sample to the clinical application. This is where I'm going to end. I'd like to thank our team within the CSIR as well as ReSyn Biosciences and all the collaborators and research partners within South Africa and overseas. And lastly, I'd like to thank you for your time and let you know if you have any questions.